Hello everyone and welcome to the biology lecture series, and today we'll be discussing and we'll be sharing the topic transportation. Okay, so in the previous lecture we have already discussed about the transportation in the higher plants, where we studied about the transpirational cohesion theory, also known as transpirational pull theory. Then we studied about the essence of sap, how the uptake of water takes place in the plant. And then we also studied about the vascular system. Then we also studied about the transportation in humans. Okay, so there blood was the carrier, and then we studied about the blood and the functions of the blood. All right, and in this session we'll be studying about the rest of the topics. So in this session we will cover how a blood clot is formed. So we'll study about the blood clot, okay, formation, and then we'll study about the rest of the topics. All right, so in the beginning I will give you a brief introduction about the blood clot, and then we will study about the blood groups, how many blood groups are present in the humans, and which blood group is the universal donor and which one is the universal receptor. All right, we'll study about all these. So let's start with the blood clot here now. So blood clot, what is blood clot? Blood clot is an conglomeration of the platelets. Means you can say grouping, a kind of a grouping of the platelets, proteins, and the cells in the blood that adhere together. So this is known as the blood clot. So blood clot, blood platelets, these are involved in the blood clot formation. We have studied in the previous lecture about the roles of the blood platelets. So these blood platelets are involved in the formation of this blood clot. Now, whenever an injury occurs, the body forms a blood clot to halt the bleeding. Okay, so what is the role of the blood clot? Why blood clot is formed? Blood clot is formed to stop. Halt means to stop the bleeding. Otherwise, there will be continuous bleeding and the organisms can die because of excessive bleeding and loss of the blood. Now, following the cessation of the bleeding and the completion of the healing, the body typically disintegrates and eliminates the blood clot. So first blood clot is formed, and then when the skin is healed, then the body eliminates this blood clot. Alright, so we'll study about the formation of the blood clot in detail today. So now this is the process, okay, or these are the steps that are involved in the formation of the blood clot. Okay, so you can write here. These are the steps. First, I am telling you the general steps. Okay, so these are the general steps that are involved in the formation of the blood clot. First is the platelets. First, they form the plug. Okay, first is the formation of the plug. So when this plug is formed, now the platelets aggregate to form a plug in response to the triggers released upon the blood vessel damage. So whenever there is a cut or whenever there is some injury, then the blood vessels, they get damaged, and the damage of the blood vessels, it triggers the response, and in this response what happens, the blood platelets, they aggregate to form a plug. Now, adhering to the damaged vessel walls and to each other, they undergo shape changes to create a plug that seals the breach, preventing the blood leakage. So what happens, when this plug is forming, then there is a change in the shape of the blood platelets. All right, and this change helps in the formation of this plug, and this plug, it seals the breach, or you can say it seals the injured area. Okay, and thus it prevents the blood loss. All right, so first step is the formation of plug by the platelets. Next is then the clot grows. So this is the second step, growth of the clot. Now the clotting factors, we'll talk about these clotting factors in the upcoming slide. So these clotting factors, these are the proteins within the blood. So blood we have already discussed. It consists of cellular component and acellular component. So cellular components were, if we talk about the blood, first of all, it consists of the plasma. Plasma we have already discussed. It is around 91 to 90% water. All right. Apart from this water, it also contains some inorganic molecules, inorganic substances, then also contains proteins such as fibrinogen, which is involved in the formation of the blood clot. Then the other components are the cellular components. What are these cellular components? So these are your RBCs, WBCs, as well as the platelets. Okay, and all these RBCs, WBCs, and the blood platelets, they are part of the plasma. Okay, they float in the plasma. Sorry, they're not the part of the plasma, they float in the plasma. Okay, now the clotting factors and the proteins within your body, they communicate with each other to indicate a swift chain reaction. Now a chain reaction is stimulated. Okay, this sequence culminates in a dissolved substance in your blood, transforming into elongated strands of fibrin. So in the end, fibrin is formed. We will see how this fibrin is formed. So a chain reaction is now stimulated, and this sequence culminate in a dissolved substance. So this is fibrin is a dissolved substance present in the blood. Okay. So this dissolved substance in your blood is transformed into this fibrin. We will talk about this dissolved substance. What is this dissolved substance? Which is transformed into fibrin, which are elongated strands. Now these fibrin strands, they intertwine with the platelets, which are present in the plug. Okay. So you can say fibrin, they intertwine with the platelets present in the plug, forming a mesh that ensures the additional platelets in the cells. Okay, that combines with the additional platelets in the cell, and consequently the clot strengthens significantly, becoming more resilient and enduring. Okay, so this is known as growth of the clot. The next step or the final step is the body breaks down this clot. Now the clot is formed, the wound is healed, and after that you have to eliminate this blood clot. Now once the damaged tissue undergoes healing, the clot becomes unnecessary. The resilient fibrin, okay, so end product is fibrin. All right, this fibrin strand, they dissolve, allowing the blood to reclaim the platelets and cells comprising the clot. Okay, so these were the 3 steps. First was the formation of the clot with the help of platelet formation of the plug. Okay, here platelets play a role in the formation of the plug. Second is the growth of the clot. Okay, growth of the clot, we have seen how the clot grows, and third is the elimination of the clot. So these are the general steps or the general process involved in the blood clot formation. Now we'll study about the clotting factors and the entire process, how this clot is formed. Now blood clotting again. Now at the site of the injury of the blood vessel. So the injury of the blood vessel is the trigger for the formation of the blood clot. Now at the site of the injury of the blood vessels, the platelet induce the blood coagulation through the release of thromboplastin. So thromboplastin is also known as thrombokinase. So this is a kind of an enzyme. So first, there is a release of thromboplastin, also known as the thrombokinase. All right. Now, this thromboplastin, what is the role of this thromboplastin? This thromboplastin, we have already discussed in the previous lecture. It changes the prothrombin, which is present in the blood plasma, into thrombin. Okay. So these are the enzymes. Thrombin is an active enzyme. So here you can see blood clot formation is a multi-step process. It is a multi-step process. All right. Now, this thrombin, what is the role of this thrombin now? This thrombin then converts the soluble protein. We were talking about the soluble protein. The soluble protein is known as fibrinogen. So this fibrinogen is then converted into insoluble fibrin. Okay, so this fibrin is the end product and this is present in the clot, or this forms the clot. So it is clear, whenever there is an injury, then what will happen? It will trigger the synthesis of the thromboplastin. So thromboplastin is also known as thrombokinase. So this is an enzyme which then further converts the prothrombin into thrombin. Now prothrombin is converted now into thrombin. Now what is the function of the thrombin now? Now thrombin converts the soluble protein fibrinogen into fibrin, and then fibrin then it interacts with the other cells and it forms the blood clot. Now fibrin, it forms a network which entangles the RBCs, the blood platelets, to form the plug, also known as the clot over the injured area. Okay, now fibrin is formed. Fibrin later on interacts with the, or entangles with the RBCs as well as the blood platelets, and then a plug is formed or a clot is formed. So clot consists of what? You can say clot consists of this fibrin protein. So this is an insoluble protein. In addition to this, it also consists of the RBCs as well as it also consists of the blood platelets. Okay, so these together forms the blood clot. Now blood clotting is usually completed within 2 or 3 minutes. So it is quite fast. Otherwise you can lose a lot of blood. Now this is then flow chart. These are all the reactions that are going on in the formation of the blood clot. The first, injured tissue is the trigger for the formation of the blood clot. So here, injured tissue, blood platelets, they get activated and they help in the formation of the plug. So blood platelets, they arrive at the site of the injured tissue. Okay, and then what is released? Thromboplastin, also known as thrombokinase, this is released. Okay, so this is the first enzyme, you can say. Okay, what is the function of this thromboplastin? This thromboplastin then converts prothrombin into thrombin. So here you can write, so this, I'll write it in English. Here the enzyme is thromboplastin. Okay, now thrombin is produced. Thrombin is also an enzyme. Okay, so this is the activated form of the enzyme. Now thrombin will then convert, here you can write thrombin. So this is the second enzyme. Now fibrinogen, which is a soluble protein, is converted into fibrin, which is an insoluble protein. Now what happens? This fibrin, then it entangles with the red blood cell as well as the blood platelets. All right. And then a clot is formed. All right. Then the clot grows and then finally the clot is eliminated by the body whenever the wound is healed. Okay. So this was the process of the formation of the blood clot. Now let's study about the blood groups. Okay, so blood groups, you know, already know they are of 4 types: A, B, AB and O. So Landsteiner, he discovered that the blood of different individuals, it did not match each other, but there were some biochemical differences. So there are some biochemical differences. All right. So biochemical differences means there is some difference in the presence of the antigen. Okay. We know about the Rh antigen also, and we know about the A antigen and the B antigen. Okay. So the positive and the negative, we say the blood group A positive or A negative. So positive means, okay, if you say A positive, it means it contains an antigen Rh. Okay, this is present. And if you talk about the A negative, then this Rh antigen is absent. And A blood group. So this is the A blood group. It carries the antigen A. A antigen is present. And if we talk about the blood group B, so B antigen will be present here. So Landsteiner, he discovered the antigen A and B and the blood groups ABO. So these blood group is known as the ABO system, and it is based on the presence of the antigen A and B. Now these antigens are also known as agglutinogen. Okay, so these are antigens. What are antigens? These are present on the surface of the RBCs. Okay, so they are present where? They are present on the surface of the RBCs. And these antigens, they are nothing but these are glycoproteins present on the surface of the RBCs. Now for each antigen, there is a corresponding antibody. All right, this is clear. Now, thus there are two antibodies. So antibodies are known as agglutinin. So two antibodies, so there were two antigens. So there will be two corresponding antibodies, A and B, that are present in the plasma. So antibodies are present where? They are present in the blood plasma. Now, I've already told you there are four types of the blood groups depending on the presence or the absence of these antigens. So let's have a look at these blood groups. So blood groups, there are four types: A, B, AB, and O. So if we talk about the blood group A, here the antigen present on RBCs is A. All right. And the corresponding antibody will be B. Okay. So A blood group people will have antibody B. They cannot have the antibody A. Otherwise, there will be a incompatibility. All right. And the blood people with blood group B, they have the antigen B, but the corresponding antibody will be A. All right. So if they contains B antibody only, again there will be mismatch. All right. There will be incompatibility. Now AB blood group. So these AB blood group, they have both antigens A and B, but they don't have any antibodies. That's why they are known as the universal acceptor, you can say, or universal recipient. Why they are known as universal acceptor? Because they lack the antibodies. So no antibodies are present. So they can receive blood from any blood group. They can receive blood from A, B, and O as well. So they are known as universal acceptor or universal recipient because they lack the antibody. So if antibodies are absent, there will be no incompatibility. There will be no reaction. So if A blood group, okay, if the blood group A person donates blood group to the AB person, so there is no antibody here. All right, no A antibody. So what will happen? There will be no incompatibility. Whereas if we talk about blood group O people, they don't have any antigen. So, okay, but they have both the antibodies in the plasma. So they are known as the universal donor. Why they have universal donor? Because they lack the antigen. There is no antigen present. So if there is no antigen, then what will happen? There is nothing to activate the immune system. So immune system is not activated because antigens, they activate the immune system. So if the person with the O blood group donates blood to any of these A, B or AB group, then the immune system will not be activated. It is not activated. All right. So that's why the people with blood group O, they are known as the universal donor. Now, blood, it is a life-saving fluid. It is often needed during accidents and the operations. Now, the transfusion of blood is only done when blood group is known. So initially, if you go in the hospital and if someone needs the blood, then the doctor will first test your blood group as well as that of the patient, and only then blood transfusion can be done. Now, blood groups are of 4 types: A, B, AB, and O. Blood group O, okay, it is known as a universal donor, means it can donate blood to any group, A, AB, B, and O, but it can receive blood only from the O blood group. Okay, so O blood group doesn't consist of any antigen, but it contains both the antibodies. Now, similarly, AB blood group, they are known as universal recipient or universal receptor because they don't have any antibodies. It can receive blood from any blood group, but it can donate to AB only. All right, you have to remember it. AB blood group can donate blood to only people that have AB blood group. So AB blood group, there is both the antigens present here. All right, so both the antigens, so it can activate the immune system of the other person. Now let's talk about the blood transfusion. The transfusion of the blood from a healthy person to a patient suffering from blood loss due to injury or surgical operation. This is known as blood transfusion. Okay, the blood transfusion is you can say the transfer of the blood or the donation of the blood to the patient from the healthy person. This is known as blood transfusion. Now, blood transfusion is done during the surgery or during the injury when there is loss of blood. Now, for this, all major hospitals, they have the blood banks. So within the hospitals, there is a separate blood bank that provides blood to the patient. So in the blood banks, the blood is collected from the voluntary and the professional donor. So there are a lot of camps, blood donating camps that these hospitals organize, and these blood is stored in the blood bank. Now, before preservation of the blood is tested for its blood group as well as the Rh factor, whether the blood group is positive or it is negative. So Rh factor is also a protein which is present on the surface of the RBCs. All right. Now, though theoretically a patient may be able to receive the blood of 2 or more types, it is always advisable to have the donor blood of the same group as that of the recipient. Okay. So, like AB blood group, they are universal recipient, but it is advised that they get the blood only from the same group, means AB only. Now, rather, the blood of the donor is always cross-matched before transfusion to exclude any chance of incompatibility. So this is a precaution that is taken. So the blood group matching is done, or you can say the blood of the donor as well as the receiver, it is always cross-matched. All right, before the transfusion is done. So this is a precaution that is taken. Otherwise, what will happen? If the blood gets mixed, if the A blood group donates blood to the person with the B blood group, then what can happen? Cross match can happen, incompatibility will occur, and the person may die also. Okay. Now, when blood from the donor is added to the blood of the recipient, it is necessary to avoid bringing together corresponding antigen and the antibody. Okay, so if antigen is A, its antibody will be B. All right, so we have to avoid this. Otherwise, there will be incompatibility or mismatch. This is also known as agglutination. So these two can combine together and this will lead to incompatibility, or it can cause the agglutination reaction, and because of that thick clots of blood can form or clumping of the RBCs can happen. Okay, so this causes the clumping of the RBCs whenever the antigen and the antibody, if they react with each other and if they are of the corresponding type. All right, means A antigen is present and B antibody is present. Now, does the antigen A in the RBCs of the A individual react with the antibodies of plasma of group B individuals? All right, this is clear. So a person with the A blood group, this will, okay, if this is the blood group, then it can react with the antibodies of the plasma of the B blood group. All right, this is clear. Now this phenomenon is known as agglutination. Okay, where the antigen of the A in the RBCs, so here the antigen is A, it can react with the antibodies of the plasma of group B individuals. All right, this is clear. So B individual, all right, means the antigen here is B. Okay, and here the antibody will be A here. All right, so here it should be A. Okay, A person, this will have the A antigen. B, this will have the B antigen. Okay, now here a mismatch can occur between the A antigen and the B antibodies. So B antibodies will be A. Okay, so person with B blood group will have A antibodies and these can react with each other. And we have to avoid this. So this should be A, not B. Now, let's discuss this table. So this table shows you the human blood groups and the transfusion. All right. Now here, the blood group of the donor is given. Here the blood group of the recipient is given. So the people with the O blood group, these are the universal donor. They can donate blood to O, A, B and AB. We've already discussed why they are universal donor. Then people with A blood group, they cannot donate blood to O. They can donate blood to A as well as AB. People with B blood group, they cannot donate blood to B. They cannot donate blood to A, but they can donate blood to B as well as the AB. Okay, so B antigen is present here in the B also, and AB also contains, whereas A antigen, this is present in the A as well as the AB. Now, if we talk about the AB blood group, so AB blood group can only donate blood to the AB individuals. So tick marks means the compatible, cross means the incompatible, RH factor. Okay, if we talk about the RH factor, it determines the positive and the negative sign. Now the RH factor which is present in the blood can be genetically determined also. We can determine it. Now most of the people, more than 85% are RH positive, while a few are RH negative. Both people lead a normal life. Now this is the case here. You have to concentrate here. If an RH woman marries with an RH positive man, then its pregnancy is normal. But the second pregnancy, what happens? The baby can die in the second pregnancy. But there are now techniques and procedures that can save the child. So what happens here? Initially, if an RH negative mother, it marries an RH positive father. So there are 50% chances that the baby can have RH positive blood, it can have RH positive protein present on the RBCs. So in the first pregnancy, it is okay. All right, because there is no mixing of the blood. But during the birth, of the first baby, there can be mixing of the blood. Okay, there can be mixing of this blood. So what happens? The mother is exposed with this Rh antigen. Okay, and the mother, okay, it gets exposed to the Rh positive protein of the baby during the birth, and then mother can develop the antibodies against these Rh positive. Against what? Against the Rh positive factor. Then what happens during the second pregnancy? During the second pregnancy, if the baby is again Rh positive, then what can happen? The mother already now antibodies are present in the mother. So the antibodies, AB means antibodies which were produced against the Rh positive, these can react with the Rh positive of the baby. Okay, so there are chances that the second baby can die because it also contains the Rh positive protein, and the mother have antibodies against this Rh positive, and that antibodies developed during the mixing of the blood during the first pregnancy. All right, so this can happen. All right, but there are some certain techniques that can help in that can help to save this baby. So some of the techniques are like this: you give the mother injections of a protein known as ROGAM. So ROGAM, these are just globin proteins, okay, and these are given to save the child. All right, so this was the end of this session. We'll talk about rest of the topics in the next lecture. Thank you very much for listening.